Hey guys, this lesson is on proteins. Proteins have carbon, hydrogen, and oxygen, just like the other two we've talked about so far. But they also have nitrogen, which is a little bit unique compared to the other ones. Their job is to help with movement, to provide structure. They're part of our cellular components. They act as messengers and receivers, especially when we talk about hormones and how our body reads messages that come through. Also in defense against disease, part of our immune system, and then with biochemical control. Examples are keratin, that's in our skin, in our hair, on our nails. Actin and myosin are another example. That is the protein that is found in your muscles. Okay, so actin and myosin is found in your muscles, which again help with movement. And then you have antibodies. Hopefully you've heard of that. That's in your immune system, so they help us fight off infection. And then one of the most important functions of proteins is the biochemical control, which is controlled by enzymes. Okay, we will have a separate video on enzymes because they are so important. All right, the monomers, or the building blocks for proteins, are called amino acids. Okay, amino acids are the building blocks for proteins, and I'm going to draw that for you. Okay, so it starts out as a carbon in the center, and then to one side you have a carboxyl functional group. Okay, so this is a carboxyl group. Oops, that's a YL. Carboxyl group, and then to the other side is an amine group. This is NH2. Okay, so this is your amine group. But remember that carbon has to have 4 bonds, okay, to be stable, so we need one here and one here. I'm just going to put a hydrogen here, and then down here is my R group. Okay, this carboxyl group over here makes this molecule acidic. And this is just, when it has a nitrogen in there like that, the NH2, you know it's an amine group. Okay, so we call this whole molecule an amino acid. Okay, so an amino acid. So every single amino acid has a group that looks like this and a group that looks like this attached to this central carbon. Okay, so that's an amino acid. Well, there are 20 different amino acids. Okay, so if every single amino acid has the amine group and the carboxyl group, the only way we can tell the 20 different amino acids apart is by this R group. Okay, so that means there are 20 different R groups. Okay, so that's 20 amino acids, which are different based on what we put here. So this is kind of like a variable in math. Okay, it can change. Okay, but these two do not change. All right, so what if we wanted to bond amino acids together? Amino acids have a special bond when you bond them together. So let's just simplify it. Say here's an amino acid and here's an amino acid. The bond that forms between them is called a peptide bond. Okay, that's a peptide bond. Well, if I want to make a whole bunch of peptide bonds and combine a whole bunch of amino acids together, then I'm getting a whole bunch of bonds. Okay? When I get this, I no longer have individual amino acids. I now have a polymer, which we oftentimes refer to as a polypeptide. Okay? So sometimes a polypeptide is used instead of saying a protein, but they're really the same thing. This one is called a polypeptide because it has many peptide bonds. Okay? Now, to show you how these amino acids really bond, I'm going to show you this picture here. Okay, so this is an amino acid and this is an amino acid. Okay, and you can see that they have just formed a peptide bond. Well, what do you notice was removed? If you look right here, these are the elements that were moved. You had an oxygen and 2 hydrogens removed. Well, that's water, H2O. Okay, so they removed water in order to make this bond. That is called dehydration synthesis. Dehydrate, you remove water. Synthesis, you make. So you're removing water to make this bond. Okay, that is called dehydration synthesis. Well, what if I wanted to go the other direction? What if I wanted to take this protein and break it down into amino acids? Well, that means I have to do the opposite. Instead of removing water to make the bond, I would have to add water to break the bond. And if I do that, I'm going to be left with my simpler molecules, my monomers. Okay, when you add water to break the bond, it is called hydrolysis. Okay, and you can do that with carbohydrates, lipids, proteins. I just use proteins as our first example of that. Okay, now if we go back to this slide right here, I just want to remind you that amino acids are fairly small compared to lipids. Lipids, remember, are really long. They have really a lot of bonds, which makes it have a lot of energy. Whereas the amino acid is pretty simple right here. It only has 4 calories per gram, just like carbohydrates. So carbs and proteins have 4 kilocalories per gram, whereas fats have 9 kilocalories per gram. And that's because carbs and proteins are fairly small, not a lot of bonds, compared to the lipids, which have a lot of bonds. Okay. Hopefully this is helpful for proteins. Our next video will be on enzymes, which is an example of a protein, which are very important.